All right, this will probably be a little bit clunky, but we're going to attempt it anyway. We're going to start with the cycle of cross-bridge cycling. And remember, because it's a cycle, there's not really a single place that it starts, but we're going to start at the point that I believe probably makes the most sense. And what I'm going to draw first here is the thin filament. Oh, these should all be touching, so we'll make those touch there. Of actin molecules. So these are the individual actin monomers that form the chain of the actin filament, or the thin filament. So here's actin, and we'll draw the thick filament, the myosin, in blue. And so here is the myosin. Here's the myosin head, currently not touching the myosin filament, and it has ATP attached to it from the previous round of cross-bridge cycling. And that's where we're going to begin. And what's going to happen here is that ATP is going to be hydrolyzed, and then the myosin head is going to be recocked. When the ATP is hydrolyzed, the myosin head is going to move back into this position. I'll draw it in dashes. It's not yet bound to the actin, but it has recocked into a position where it can move over and bind to the actin. The energy from ATP hydrolysis has not yet been used. The energy was not used to recock the myosin head. That was a conformational change that just happened because of the ATP hydrolyzing. So the energy is stored in the myosin head, and we will use it later. All right, so here's the myosin head recocked, and ATP was hydrolyzed, so we still have ADP and the phosphate, which gets a little I for inorganic, meaning there's no carbon associated with it. The ADP and the phosphate group are still attached to the myosin head under this condition. Now what's going to happen is this myosin head is going to now move across and bind to actin. Actually, let me have it bind to this one here. And that is our next step. So now the myosin head moves over and binds to the active site on actin, which, remember, has already been exposed because the calcium is bound to troponin, and tropomyosin has moved away from the active site. It is no longer blocking it. Otherwise, this would not be happening right now. Okay. The ADP and the phosphate are still attached. This is really now formation of a cross-bridge. And what's going to happen is that the sequence is actually a little bit debated, but that's really not important for our purposes. The ADP and the PI are going to be released kind of as the myosin head now pulls in this direction. And so the myosin head is going to move over into this position, remaining attached to the actin. And so the whole actin filament, as a result, is going to be pulled this direction, which, of course, pulls on the Z-disc, pulling everything closer together. Again, because this happens millions of times across and down a muscle, it ends up with muscle contraction or more muscle shortening. All right, that brings us to this position where the myosin head has undergone the power stroke, pulling on the actin and releasing the ADP and the inorganic phosphate. And that was the pulling that we just talked about. Now, it's going to remain in this position for some time. Well, I said that incorrectly. Not that it happens forever, but it will remain there until another ATP comes and attaches. When ATP comes and attaches again to the myosin head, then the myosin head will detach. So in this view here, the ATP has come and attached to the myosin head, and the myosin head detaches with ATP attached. If you go back to the beginning of the video, this is where we started. The head of the myosin head had ATP attached and was not attached to the actin filament and needed to be recocked. Now remember, it's going to recock in this direction when this ATP hydrolyzes to ADP and phosphate once again. Now my writing might not be too clear here, but this myosin head has recocked, moving in this direction. I've probably drawn it just a little bit close here. Let me remedy that. It's really not that close because it's not attached right now. And the ADP and the phosphate are attached. And now from this position, it can come up and form another cross bridge. Remember before it formed the cross bridge with this actin down here, this one. Now it's going to form a cross bridge further down. It may not be the very next actin molecule as I show it here, but it's going to form another cross bridge further down, and then it can undergo the same cycle and it can pull again. This is known as cross bridge cycling. So this is known as cross bridge cycling, and as that myosin head continues to reach down and grab another actin molecule and pull on it, it kind of, if you will, walks along the actin filament. And so this is known as the walk-along theory of muscle contraction. Now, because there are millions and millions of these in a muscle that are doing this, not all of them do it in synchrony. You have some that will be holding on to the actin, some that are let go, and each one is reaching up and grabbing and pulling again throughout this cycle. Now remember, during this process, the thick filament does not move. It does not change length. The thin filament moves, is pulled past the thick filaments, but the thin filament also does not change length. What it does is pull on the end of the Z discs, pulls them closer together, resulting in contraction. And so this is part of what we then... This is then what we call the sliding filament theory, because the filaments are sliding past each other. And if you want some additional information on how this all works, if you really feel like animations and drawings help you to understand this a little bit better, go back into the PowerPoints, because there's some animations in there that may help. I'd also do an exploration of YouTube, as there's a lot of good videos out there on YouTube as well.